what I'd like to do today is talk to you about life in the slow lane. If some of you think your experiments take a long time, trust me, ours can take an awful lot longer. And I'm going to give you 4 examples of very long-term experiments that we've carried out, and then talk in a bit more detail about what we think the underlying mechanisms are as to why Antarctic species are so slow at anything they do. So the first example are heated settlement panels. That's shown here, and these are a derivation of the standard settlement panels you put in the water that are colonized by encrusting species. But these ones are modified, so they're linked up to an electricity supply on the surface, and this enables us to heat the surface, the boundary layer of the settlement panel, to +1 or +2 degrees above ambient temperature. And these can then be put in the marine environment, and they can stay there for months, and you can monitor them by sending divers down and photographing them. And it means that you can conduct temperature experiments in the natural environment with all the food supply, the temperature and the nutrients in the natural environment, as opposed to the kind of very artificial lab experiments. And you can carry out these very long term. So we put some of these panels just off Rothera base in the Antarctic, and we looked at the results at 9 months. The reason we chose 9 months was actually this was the end of the summer season, and unfortunately a big iceberg came along and bashed our main site. We actually had 3 sites out, but our main site was actually more or less devastated. And Gail Ashton, who was in charge, the postdoc in charge of the project, managed to rescue the plates, and we decided that then maybe we'd better analyse them in case anything else happened in winter. And she found some amazing results. So this picture here shows a panel under normal ambient conditions, so at 0 degrees, and this is just +1 degree of warming. There was a phenomenal increase in growth rate, so the growth rates almost doubled in a wide range of species, which is phenomenal given that you've only increased the temperature, or we'd only increased the temperature by +1 degrees. This meant that you got a real dominance of pioneer species and a decrease in diversity and evenness. So at 9 months, your results would say, well, this is fine. These animals can all survive with +1, +2 degrees of warming, so they're going to be fine under climate change. The only question we had really was that these guys are filter feeders, and this was at the end of the summer, so at the end of the phytoplankton bloom, when food was really plentiful. And we did wonder what would happen in the long, dark Antarctic winters, whether these animals will be able to survive without any food and then maintain these growth rates the following year. Fortunately, we had 2 other sites that were still going strong, and we managed to rescue some of the plates that were still working, although we'd lost all the ledges that we put them on, into the aquarium at Rothera, which is our base in the Antarctic, and kept them over the winter. And this enabled Lara Villota Nieva, a PhD student at BAS, to go in the following summer, and part of her project was to study the molecular response of these guys, spirorbid worms, on the Antarctic panels. The first question we had really was, have these organisms acclimated to +1 and +2, because their growth rates were so phenomenal. And so the easiest thing to do to start with really was to look at their upper lethal temperatures. And this is a really useful metric to understand if animals have acclimated at the whole animal level, because if they've acclimated to the warmer temperatures, their upper lethal temperatures will increase. As you can see here, we have a graph of upper lethal temperatures, and you can see it's fairly obvious that basically the more we warm the animals up, the lower their upper lethal temperature goes up. So these animals really don't seem to be looking to be in terribly good shape. And this was validated by the molecular results. And basically, when we looked at the differential expressions, a huge amount of differential expression on +1 panels between control and +1 animals. So about 14,500 genes upregulated, which, to be honest, if these animals had acclimated, you would expect no difference at all. But 14,500 was pretty major. it was all sorts of different functional groups, including a lot of the stress proteins. So clearly the animals were not surviving terribly well, and they were upregulating absolutely everything they got to try and adapt to the new conditions. When it came to the animals at +2, there were far fewer genes differentially regulated, so only about 1,000. But when we looked at these were mainly genes to do with, and that's shown in this diagram here, transcription and translation, the cell cycle, energy production, and the cytoskeleton. So really the essential genes they needed to keep the cells running, and they basically turned everything off, including all the stress proteins, although clearly they were obviously very stressed because their upper lethal temperatures had gone down. And so what we concluded from these results was that these animals, although they had this massive growth rate at 9 months, they were clearly couldn't cope long-term with the chronic temperature increase and were in a phase of terminal senescence. And therefore, if you look to the results at 9 months compared to 18 months, you've got a very different story. So at 9 months, you would think the animals would do really well under climate change and everything was fine. But if you left it for another 18 months and across the Antarctic winter, the long-term prospects for these organisms really aren't very good at all. So a completely different picture depending on the timescale of your experiment. But this does depend on the animals you look at, and we got very different results when we looked at a scavenger. This is the Antarctic sea urchin, Sterachinus neumeieri, and this was a PhD student, Colleen Suckling, who worked in our lab, who cultured Antarctic sea urchins for 2 years in conditions of ocean acidification and warming, so a combined stressor. So she kept animals and there was a low temperature, so that was ambient 0 degrees. The high temperature was actually, it was only +2, which doesn't sound much to you, but it's a pretty big deal to an Antarctic species. And then +2 degrees with moderate acidification and +2 degrees with a kind of more severe acidification treatment. And the reason we kept these animals, wanted to keep these animals for 2 years was obviously long-term experiments and good things, but these animals take 2 years to develop, for their eggs to fully develop. So although they spawn every year, the eggs that are produced take 2 years overall to develop, and we wanted to see what the effect of ocean acidification and warming was on egg development and how that would affect their offspring if those eggs were fully developed in the altered conditions. So these data here show oxygen consumption of the adults as a proxy for their metabolic rates. These data just show the high temperature, so the +2 degrees in black, and then the 2 ocean acidification conditions. And you can see when the animals are first put in, the ocean acidification adds a bit to the stress, so the metabolic rate goes up. But gradually, the difference between the different treatments decreases as time goes on across the 24 months. And if you do all the statistical tests, you basically find that by 8 months, there is no statistical difference between any of the treatments. And if you're using metabolic rate as a proxy for acclimation, these animals have clearly acclimated to the altered conditions, but it's taken them 8 months to do so. Again, with these Antarctic urchin experiments, we used 2 timescales. So we looked at 2 different time periods, and again, similar to the heated settlement panel results, we got very different results depending on when we looked at these animals. So these data show egg size and the results at 6 months. So the black column this time is actually eggs at 0 degrees, developed at 0 degrees. Then we have +2 degrees followed by the 2 ocean acidification conditions. And you can see that at 6 months, the eggs under the ambient conditions are clearly much bigger than the others. However, at 17 months, the situation is completely reversed, and the animals that are in the warmer, more acidic conditions are actually statistically significantly larger than those under normal conditions. And this is a well-known phenomenon. It's been observed in quite a few species that when conditions are less favourable, the adults produce fewer but larger eggs to try and increase the success rate of their offspring under the kind of more severe conditions. And this was shown through to the survival of the offspring. So at 6 months and 18 months, Colleen cultured the offspring for about 17 days. And the top graph at 6 months shows the low temperature control, so the 0 degree animals, about 85% of them survived after 17 days. It was a little lower for the animals at +2, which was 70%, and then really the animals under the warming and acidification conditions weren't doing particularly well at all. And so you would actually say that if you just took these results at 6 months, that in the future, under warming and acidified oceans, these animals really wouldn't do particularly well at all. However, the results change very dramatically when you do look at them at 18 months. So, and these are the data shown in the second graph here. And the high temperature control, so the +2 animals, have increased their survival rate from 70% to 95%, which is pretty good going. And more dramatically, the altered pH and warming conditions have also improved. So the severe conditions from 30 to 75 and moderate from 50 to 75, from 30 to 50%, which is very dramatic. And obviously these animals are in a much better place than they were 6 months ago. And therefore you'd say that by doing the experiment for 2 years, it gives you a better idea of how these animals would survive under altered conditions, and clearly it would appear that they would survive fairly well, especially when you consider that we put them in these altered conditions really, really quickly, and we weren't working on climate change timescales. You may be thinking that I haven't actually mentioned the 0 degrees animals in the second graph, and unfortunately they're kind of down here. And this is one of the issues you have with long-term experiments, that at some point something will go wrong. And we think something went a bit wrong with the 0 degrees control, so we're not really sure what's going on there, but we don't think that, it's just one of the things that you kind of have to live with. And it's always very difficult to say when you're doing experiments for about 2 years. So these 2 experiments show you the very different results you can get by looking at the same experiments, but using different timescales. And what I'd like to do now is look a bit more in more depth about potential mechanisms as to why some of these responses may be so slow. So this third example is actually limpets, and these occur in the Antarctic, in the intertidal region, down to about 100 meters. And the intertidal animals are a very different shape to the subtidal animals. So they have a much thicker shell and it's much pointier. Obviously it's much thicker because they have to survive all the brash ice in the intertidal region in the Antarctic. The subtidal animals have a much shallower shell, and it's actually much thinner. And it was thought for many years that these were 2 different ecotypes. That they're not. I mean, these guys are broadcast spawners and they are a genetically homogeneous population. We did do the analysis. And the difference in shell weight is actually fairly dramatic. It's this graph here shows you the dry shell weight of an intertidal animal, and it plunges dramatically to the subtidal animal. So being exposed to brash ice does dramatically increase your shell thickness. And as Antonio mentioned, one of my interests is in biomineralization, and particularly in biomineralization in mollusks. And I was fascinated by the fact you got these 2 very different thicknesses of shell. And I thought, well, maybe we could turn an intertidal animal into a subtidal animal. Maybe we could start to play around with biomineralization mechanisms in these animals. And so this started a whole series of experiments that probably went on for about 5 years in total, looking at the response of these animals to the intertidal region. So we decided to start off with a transplant experiment. So what we did was we took animals from the intertidal region, painted them yellow, put some of them back as controls, then moved some of them down to 15 meters which is shown here. And then we got divers to collect limpets at 15 meters. We painted them white, put some of them back as controls, and then moved some of them up to the intertidal region. Now, having said that we wanted to do long time scales, the idea was we would leave them there as long as possible and then, go back several months later and collect them if we could. But even though these limpets weren't homing limpets, they started to move back to where they came from. So we had to finish the experiment after a month, because otherwise we were going to run out of transplanted animals. They just, even though we put 60 transplanted ones back in each site, they were disappearing fast. Now, we already knew that there were very different expression profiles between intertidal animals and subtidal animals, and I'm a molecular biologist, so all the analyses we do are molecular based. And in general, the intertidal animals had a very stressed gene expression profile. They were expressing a lot of stress genes permanently, because these animals are regularly exposed to air, they're desiccated, and in the intertidal region of the Antarctic, it can get up to +6 degrees, which is pretty warm for these guys. So, and what they do is they permanently express stress proteins at a fairly low level as a kind of preparative defense, because it would appear that that's far more energetically economical than having to up and down regulate the same genes on a regular basis. When we looked at the gene expression profile of the subtidal animals, it was very much a kind of, there were no stress proteins at all. They seemed perfectly happy, they're eating, reproducing, whatever. So you had very different expression profiles. Now, when we looked after a month at the transplanted expression profiles, we got quite a surprising result. So when we looked at the response of the intertidal animals that were transplanted into the subtidal region, the gene expression profiles hadn't really changed. It was still very much this kind of stressed intertidal profile. However, when we looked at the expression profile of the subtidal animals moved into the intertidal, they were starting to express the intertidal profile. So they were starting to produce biomineralization genes, stress proteins, because clearly they needed to compensate for desiccation, warming, and also build tougher shells to survive the brush ice. So we kind of thought, well, what's going on, and why is it just one way, and why is it such a difference still after a month? So what we decided to do was a common garden experiment. So we completed all that set of experiments, but then we decided to go on and do this common garden experiment. So we got animals collected from the intertidal and the subtidal region in the Antarctic and brought back to Cambridge, where we put them in the same aquarium tank for the 9 months. Now, the reason we chose 9 months was that previous work we'd done in the lab showed these animals take 9 months to acclimate to +3. So we kind of figured that if they were just acclimating to an aquarium tank, 9 months would be more than enough. I think, how wrong could we be? Anyway, so we got some really interesting results after 9 months, and what we found was that although when we compared the expression profiles of the original intertidal and subtidal animals, they were much more similar than they were previously, so they're becoming much more homogeneous, and clearly the animals were acclimating to the aquarium tank, it still wasn't complete. We didn't really get any strong gene networks associated with the subtidal animals, but the intertidal animals, there was still the hint of this intertidal profile. And we had one gene network in these animals which showed a prevalence of stress proteins. So we had catalase involved and glutathione genes, which were all antioxidants. And so it's very clear to us that although these animals had very distinct gene expression profiles associated with a particular habitat, when you took those habitat conditions away and those individual stresses of those habitats away, the animals did start to lose those profiles, but it was a very slow process. And in terms of the inter-tidal animals, which were regularly bashed around by brush ice and exposed, the last thing they did, or the last things they erased, were these stress proteins. So they were really keeping these stress proteins hanging on as long as possible as a kind of defense mechanism, emergency defense mechanism, should an iceberg ever happen to come along. And the question was, why was this what was going on? And I think the obvious answer is epigenetics. And we were lucky enough to get an award to work with the NERC Biomolecular Analysis Facility in Sheffield to look at methylation in these animals. So we were able to do methylation sensitive amplified length polymorphisms on these guys. So when we looked at the methylation patterns of the intertidal and the subtidal animals, they were very different. And clearly that's what we'd expect, because we were hoping that methylation would be setting these expression profiles. When we looked at the animals in the aquarium tank, there were no methylation differences between them. Now, this isn't particularly surprising, because we were only looking at about 240 loci with the methylated AFRPs. Whereas obviously we were looking at thousands of genes with the gene expression profiles, so the gene expression data would be in far more detail. But what was fairly clear was that there's a really nice association of methylation associated with setting gene expression profiles according to habitat, and that this process was reversible, but it could take 9 months to take the methylation tags away from those genes. So it was potentially a very long process, and if conditions suddenly changed, then the genes that then needed to react to those conditions were methylated. It could take them a long time and predicate their responses to change. And I think that's potentially what is going on with some animals in the Antarctic. So, and that is the final example I'm going to talk about, which is aging in clams. So these guys are Latinula elliptica. They live till they're 37 years old. We can age them because they have annual shell rings. And this is a middle-aged animal. You probably wouldn't realise it, but it's about 18 years old. And this is a pre-reproductive juvenile, about 3-4 years old. And we know there's been a lot of work done both in our lab and in GeoMar in Kiel, and also in the RV in Germany, on the effects of age on these animals. So we know that thermal resilience decreases with age. The older animals have a lower immune capacity, they burrow less rapidly, they're more affected by sedimentation and injury. And at the gene level, the older animals are much less responsive when conditions change. They don't do very much. And the younger ones have a much more active stress response. So they seem to actively manage the stressful situation rather than the older ones who just seem to just sit there and do not a lot. And this was particularly obvious in a series of experiments that was run by Vicky Slight, who was a PhD student in our lab. And Vicky was working on the other area I'm interested in, which is biomineralization, and she was looking at biomineralization in the Antarctic clam. And her experiments were based on making very small holes with a modelling drill in the clamshell. These gradually healed over, so you get a tannin layer, and then that's filled in with calcium carbonate crystals. And by sampling the mantle tissue under that hole over a timescale, we hope to uncover genes that were involved in biomineralization. And in actual fact, Vicky has just published the first gene regulatory network for an Antarctic bivalve, well, in actual, a bivalve full stop, in bioinformatics. The biomineralization story is still ongoing. It's still the subject of more analyses. We just keep adding stuff to it. But the first cut results were really interesting. So this graph shows the number of differentially expressed genes against time. And we had 3 different cohorts. So we had juvenile animals, which were incredibly small. So these were less than 1 year old. And basically there was no difference. We drilled holes in them, there was no differential gene expression. And we thought, that this was probably because these animals repaired their shells really quickly. They had very thin shells, they were growing rapidly for an Antarctic animal, and they were producing shell as quickly as they could. So if you stuck a hole in it, it didn't really make any difference. They couldn't make the shell any faster, so there was just no difference. When you look at the adolescent animal, there was an increase in gene expression for about 2 months, and then everything kind of gradually went back down to normal. And these animals can take about 2 months to repair their shell. So clearly there's an upregulation for shell repair and also for potentially to fight infections. The animal was now open to the environment. But once that hole was sealed, then everything kind of went back down to normal. But the older animals, everything just kept on going up and up and up. And these guys can take at least 6 months to repair a small hole in their shell, which is pretty slow going. And why was this? So I think it's partly related to their lifestyle. So these animals live permanently buried in the sediment in the Antarctic. And as I said, they can live for 37 years, so they can live for 37 years in the same place and not move. They reproduce, they respire, they metabolize. But what I think is happening is that as they get older, they're gradually turning off all the peripheral genes that they don't need anymore, potentially by methylation. And so when conditions change, they have to kind of re-energize these genes, they have to demethylate them and get them going again. And I think this relates back to the Limpet experiment, that this process of demethylation could take a very long time indeed. And we're lucky enough just to have got an award to work with Edinburgh Genomics to do some bisulfite sequencing on young and old animals, really to look more in detail about methylation patterns and hopefully be able to associate those with particular genes. So those are 4 examples of very long timescale experiments, in terms of the actual timescale of the experiment themselves, but also we just keep adding things on to other experiments. But I think it's fairly obvious that Antarctic species can take a very long time to respond to changing conditions. And really, if we want to look at this in the future, we've really got to do 9 to 18 months duration experiments, which is not inconsequential. But what we also need to do is understand their ability to respond to change. So it's really looking at their capacities and how things like feeding mode, which affects the available energy these animals have to be able to respond to change, and also things like methylation and age differences, so how methylation affects their potential responses to change. But these animals are very slow anyway. They don't respond, they don't particularly fast at all. So the question is, why are they so slow in the first place? And you may be thinking, well, actually, when it gets cold, I slow down. I don't really do an awful lot. I mean, these animals live permanently, more or less around about 0. But there's more to it with these guys. And I'm going to briefly cover the work of Lloyd Peck, who's written 2 reviews on this subject, really, which are at the bottom of the slide. And he looked at development in gastropod mollusks, and this graph here shows the raw data. So this is brooding period in weeks compared to habitat temperature. And you can see that temperate and tropical species take about a week or so to develop, whereas the Antarctic species, there's a dramatic increase at around about 0 degrees, and they can take about 100 weeks to develop. When you convert this to an Arrhenius plot with the log of development rate over the inverse of temperature, you can see that the temperate and tropical species are all on the line, so they obey the rules for temperature, but the Antarctic species are significantly below the line. So there's something odd going on. They don't obey the rule of the temperature. When you look at growth in echinoderms, so this is the von Bertalanffy coefficient. Again, you see that the Antarctic species have a much slower growth rate compared to the temperate and tropical species. And again, when you convert this into an Arrhenius plot, The temperate and tropical species are along the line, so they obey the rule, but the Antarctic species are actually much lower than, and statistically significantly below the line. So again, not obeying the rules. Although the graph for oxygen consumption looks similar, so this is for bivalve mollusks from the tropics to the poles, you can see that the Antarctic species have much lower oxygen consumption rates compared to temperate and tropical species. When you actually plot the data here, everything's on the line, even the Antarctic species, which is a bit weird. So kind of what's going on? Why do some not obey the rule and others do? And when Lloyd combined all the data together and found various aspects that didn't obey the expected limits compared to those that did, all of these growth, specific dynamic action, duration, acclimation to temperature, egg size, they're all to do with protein synthesis and protein functioning, whereas those that fit the RNAs aren't. And he suggested that this was due to the fact that these animals have problems folding proteins at low temperatures. And this is backed up by data that's been accumulated over time on these animals. And there are other data to suggest that Antarctic species really do have problems making and synthesizing proteins. So one of the pieces of evidence is in terms of heat shock proteins. So heat shock proteins are chaperones, and what we found with Antarctic species is that when you warm them up, some species don't produce a classic heat shock response. So as you warm an animal up, the proteins start to unravel, and what happens is the animal usually would produce heat shock proteins, or the chaperone proteins, which come along and try and refold the unfolding proteins and keep everything working normally until conditions change back to normal. But some of these animals, particularly the fish and some of the invertebrates, don't produce this heat shock response. But all the animals produce extra heat shock proteins all the time, in terms of extra compared to closely related species in the temperate and tropical regions. And it's thought that this is because, although we know that it's well known that proteins denature at high temperatures, proteins also denature at low temperatures. So it's thought that Antarctic species produce extra chaperones permanently to help with this problem with protein folding in the cold. It's probably just not protein folding, it's the whole protein production thing from transcription through to degradation. So when you look at RNA to protein ratios compared to habitat temperature, again, in the Antarctic, you need much more RNA to produce the same amount of protein compared to a temperate and tropical species. This is followed through to with protein metabolism, and these data here show fractional protein synthesis rate in percentage per day of temperate and tropical species compared to Antarctic species. And this is the work of Kieran Fraser, who used to work at BAS. Now, when you look at these graphs, you can see, first off, that temperate and tropical species produce around 4% per day. But when you look across the Antarctic species, it's only 0.6. So Antarctic species are producing much less protein per day compared to their temperate and tropical relatives. But the real killer here is in the degradation and accumulation rates. So temperate and tropical species basically keep about 50% of the proteins they produce, whereas Antarctic species, they degrade 80% of what they produce. So these guys, not only are they producing much less protein per day, they're actually degrading most of it. So most of what they produce really doesn't seem to be particularly good at all. And these data are validated by data from fish. This is work of Gretchen Hoffman and Todgham in the US, who looked at ubiquitination rates in Antarctic fish and closely related New Zealand congeners. And what they found was that the Antarctic species had far more ubiquitinated proteins ubiquitin is a tag that's added to the protein to make sure it gets degraded. And the Antarctic species had far more ubiquitinated proteins compared to the New Zealand temperate congeners. So these guys are basically, they're not producing a lot of protein. What they're producing isn't particularly good. And all the evidence points to the fact that these animals have problems producing proteins at low temperatures and folding them and degrading them. Now, you might have thought that 15 million years of evolution in the Southern Ocean, these guys might have actually come up with a few answers as to how to solve this problem, and everything will work a bit more efficiently. Well, in some cases, that is true for individual proteins. So as you cool a protein molecule down, it becomes more rigid, it becomes less flexible. And this flexibility does affect the enzyme kinetics. And there are a number of examples of Antarctic proteins where there are amino acid substitutions, particularly in the catalytic site, which makes the molecule more flexible in the cold. And therefore, these proteins work more effectively in the cold compared to temperate proteins. There's also the phenomenon of gene duplication. So although the genes haven't changed that much compared to their temperate relatives, there are more of them. So they've just duplicated the genes. So effectively what these guys are doing is they're producing more rubbish protein in the hope that basically if they produce enough of it, some of it will work well enough to keep things running. And what we don't know is how widespread each of these phenomena are in the genome, because we don't really have genomes for these organisms, although the Sanger Institute has started a program of sequencing a range of Antarctic fish species, so we should start to be able to look at this in more detail in the future. But what we decided to do was really to look at whether there was amino acid bias in Antarctic fish compared to closely related temperate species. So in this case, we actually used stickleback and zebrafish because they're model organisms and there's a lot of their well annotated genomes. And our initial hypothesis was that there would be an amino acid bias in Antarctic fish, because if you look at the data from bacteria, there are very definite amino acid substitutions associated with adaptation to the cold, which improve protein flexibility. And we assumed that we would see these in the Antarctic fish. So we sequenced the transcriptomes of 4 Antarctic fish species, and these are the results. So the top graph shows the total amino acid composition of Antarctic fish species in blue compared to zebrafish and stickleback, which are the kind of yellowy ones. And there's no difference at all, really no difference in total amino acid composition, which was a bit disappointing, to put it mildly. However, Camille Berthelot, who was doing the bioinformatics, then decided to look at amino acid usage at non-synonymous positions, which is the second graph down here. And you can see that there's a really big difference between the notothenioids in kind of grey-blue colour compared to stickleback and zebrafish, which we thought was absolutely fantastic. It's absolutely great. Obviously, we got the answer we wanted. Until Camille pointed out she hadn't done all the false discovery rates, hadn't done the corrections for multiple testing. And these data are shown in a different format here. So this shows you the substitution imbalance between temperate species here, so this is stickleback and zebrafish and the amino acids involved, against the Antarctic species. The top graph is basically this the graph down here translated directly, so you can see the very big differences between the temperate and the Antarctic species. However, when you do the correction for multiple testing, false discovery, you get one result. All that difference disappears. And the really interesting thing is that all we get is the substitution of methionine for leucine. So we get methionines instead of leucines in Antarctic fish. And this is a really surprising result because methionines are a very rare amino acid anyway, and putting in a few extra methionines into a genome is really not going to make all the proteins much more flexible and work better in the cold. So what's going on? Well, there's data from other species that methionines act as redox sensors, so reactive oxygen species. And these fish survive in a highly oxygenated environment. As you cool water down, the amount of oxygen in the water increases. So these animals are in a highly oxygenated environment with a lot of reactive oxygen species. And so having redox sensors is probably quite a good move. And what we think is happening is that these animals have done the absolute minimum to alter their genomes to work in the cold. And they managed to do this because they have such a low energy lifestyle. They basically don't do an awful lot. They do it very slowly. And therefore, their cells can cope with this extra protein burden. So producing extra chaperones, a huge burden of rubbish proteins all in their cells. Now, you would think that if you warm these animals up, because they have problems folding proteins in the cold, that they would do much better if it warmed up a couple of degrees. Clearly they don't. They're all very sensitive to warming, and they tend to fall over really quickly, as I showed you with the heated settlement panels project. And so what we think is happening, although we haven't proved it yet, is that these animals have minimally modified their genomes, but they've altered a few critical proteins to work efficiently in the cold, and potentially it's these proteins that fall over when you warm the animals up. And so I think in the future, as we start to get genomes for these animals, there are a lot of interesting questions that we can really start to answer in depth by really getting to the nitty-gritty, by having full genomes, to start to really look in depth at how these animals have adapted those genomes to the cold, how they've adapted their proteins, and how that affects their abilities to respond to change, along with looking at other aspects such as methylation and how this alters their capacities to respond in the future. So that's a very quick whistle-stop tour of molecular biology in the Antarctic. So I think I'll leave it there and take any questions. I have to say thank you to a whole bunch of people who are involved in these experiments. If you have any questions and you think about them later, just feel free to contact me by email. And the pictures above show climate change in action. I first went to the Antarctic in 2006, and this is the ice cliff outside the office, and this is 2015 and 16. So even in the time I've been going to the Antarctic, I've seen big changes there. Thank you.